What if the same filter that ensures your vaccine is safe could be the key to discovering alien life? It may sound like a setup for a science fiction movie, but stick with me here, because what I'm about to share with you will blow your mind. As we speak, NASA's Europa Clipper is racing through space at 30,000 miles per hour, carrying technology to perform an initial survey of possible life on another world. This $5 billion mission launched on October 14, 2024, and is heading to Jupiter's moon Europa, a world that scientists believe harbors a vast ocean beneath its icy shell, an ocean that might, just might, contain life. But here's the thing that keeps me up at night. How do you find microscopic life in an alien ocean that's buried under 15 miles of solid ice? How do you capture something that might exist at concentrations of just a few cells per millions of liters of water? How do you ensure your equipment works perfectly in an environment so hostile it makes the most challenging pharmaceutical manufacturing conditions look like a sunny day at the beach? The answer, my friends, lies in the unexpected convergence of two fields that until recently had almost nothing to do with each other: pharmaceutical filtration and astrobiology. The same membrane technologies that remove bacteria from injectable drugs, the same quality systems that ensure batch-to-batch consistency and biologics manufacturing, and the same contamination control principles that keep our medicine safe. These are now at the forefront of humanity's search for life beyond Earth. Welcome to Apprentice.io's Industry Insights. I'm Frank Maggiore, and today we're exploring how the humble membrane filter, that workhorse of pharmaceutical manufacturing, might be the key technology that answers humanity's most profound question: are we alone in the universe? Now, I used to work for Sartorius Steam Biotech, and I started out as an application specialist in filtration technologies. One of the first support calls I ever got was from Raytheon, looking for a filter with sufficient flow and chemical compatibility for a chlorine laser. So, after that, I became the king of weird and unusual filtration projects, from end-of-the-world preppers looking for ultrafiltration units to purify insulin at home, to setting PTFE filters on fire, which is highly toxic, as part of an investigation, to developing a filter washing machine for filters that got hydrophobic chemicals on them, which prevented them from being integrity tested. And that one guy calling for assistance on his cocaine laboratory that he set up at his home address. Sure, man. During my time there, I saw images of Sartorius working with NASA and the Apollo program, where they provided the highest precision scales for weighing the moon rocks that would return to Earth. Sartorius was very proud of that history, but I thought, what if we could go bigger? What if we could be involved in one of the biggest discoveries in all of human history? And what if we could be the ones that provide the filters to achieve that task? That's got to be worth bragging rights for as long as you exist as a company. And Sartorius is already a 155-year-old company, so what's a few hundred more years of rubbing that discovery in your competitors' faces? And for those who may say that filtration technology isn't rocket science, Einstein himself worked and developed methods for the validation of filter sizes back in 1923 with his paper on experimental determination of the pore diameter in filters, which was published in German and is obviously less famous than his other works. Finding life beyond Earth isn't just a philosophical quest anymore. It's not just about radio telescopes listening for alien signals or scientists debating the Drake equation. It's becoming an engineering problem, a manufacturing challenge, a quality control puzzle of the highest order. And the stakes, they couldn't be higher. We're talking about a discovery that would fundamentally reshape our understanding of our place in the cosmos, our origins, and our future. The beautiful irony is that the pharmaceutical industry has been unknowingly preparing for this moment for decades. Every time we validated a sterile filter, every time we optimized a tangential flow filtration process, every time we developed a new virus removal membrane, we were building the toolkit for finding alien life. We just didn't know it yet. Let me blow your mind with some numbers. Europa, that ice-covered moon of Jupiter, contains twice as much water as all of Earth's oceans combined. Let that sink in for a moment. This moon, which is smaller than our own moon, has an ocean that's 60 miles deep in some places. That's 10 times deeper than the Mariana Trench, the deepest point in Earth's oceans. But Europa isn't even the most exciting ocean world in our solar system. That honor might go to Enceladus, Saturn's sixth largest moon. Enceladus is basically the galaxy's most enthusiastic geyser, shooting plumes of water vapor, ice particles, and, this is the exciting part, organic molecules hundreds of miles into space through cracks in its south pole. These cryovolcanoes, or ice volcanoes, are literally advertising the contents of Enceladus' subsurface ocean like a cosmic free sample day at Costco. NASA's Cassini spacecraft, which explored the Saturn system from 2004 to 2017, actually flew through these plumes multiple times, and what it found was absolutely mind-blowing. Complex organic molecules, the building blocks of life as we know it. We're talking about carbon chains with more than 15 atoms, molecules that on Earth are associated with biological processes. The spacecraft detected formaldehyde, acetylene, propane, and methane, evidence of hydrothermal activity on Enceladus' ocean floor, similar to the deep sea vents where many scientists believe life first emerged on Earth. Then there's Titan, Saturn's largest moon, and honestly, it's the weird kid of the solar system family. Titan has lakes, rivers, rain, and clouds. Sound familiar, right? Except all that liquid is methane and ethane, not water. The temperature there is minus 290 degrees Fahrenheit, so cold that water ice is as hard as granite and makes up the moon's bedrock. It's a world with completely alien chemistry, where the rules we know about life might not apply at all. What makes these ocean worlds so much more promising than Mars? Simple, liquid water, and lots of it. While Mars might have had oceans billions of years ago, today it's a frozen desert with maybe some salty brine deep underground. But Europa, Enceladus, and several other moons may have vast, stable liquid water oceans right now. And where there's liquid water, energy, and organic molecules, there's the potential for life. The other game changer is that these oceans are protected. They're shielded from radiation by miles of ice, creating stable environments that could have existed for billions of years. That's plenty of time for life to emerge and evolve. On Earth, we find life everywhere that there's liquid water, from the deepest ocean trenches to tiny pockets of brine and Antarctic ice. Why should space be any different? But here's the challenge that brings us back to filtration. The concentration of life in these alien oceans could be incredibly sparse. We might be looking at environments with just a few cells per liter, or even less. On Earth, even in nutrient-poor regions of the ocean, we typically find thousands of bacteria per milliliter. But in an alien ocean with unknown chemistry and energy sources, life might exist at concentrations that make finding it like searching for a specific grain of sand on all the beaches of Earth. And that's where our pharmaceutical filtration expertise becomes absolutely critical. Alright, let's get into the nuts and bolts of how filtration actually works and why the technology we use every day in pharmaceutical manufacturing is perfectly suited for hunting aliens. In pharmaceutical manufacturing, we use membrane filters for one primary purpose: keeping things out. We're removing bacteria from water for injection, filtering viruses from biological drugs, eliminating particles from parenteral solutions. It's all about creating sterility, ensuring purity, protecting patients. The gold standard is the 0.2 micron sterilizing grade filter, which removes all bacteria and larger microorganisms. We validate these filters to demonstrate better than 10 to the 7 log reduction of challenge organisms like Brevidomonas diminuta, or B. diminuta to keep it short. But now, imagine flipping that entire paradigm on its head. Instead of removing life, we want to capture it. Instead of preventing contamination, we're praying for it. Alien contamination. It's like taking everything we've learned about keeping microorganisms out and then using it to trap them instead. Let's talk about PTFE, polytetrafluoroethylene membrane filters. In the pharma world, these are our go-to filters for sterile air and gas filtration. They're hydrophobic, meaning they repel water while allowing air to pass through. They're chemically inert, compatible with pretty much any solvent or chemical that you throw at it. They can withstand extreme temperatures, from cryogenic cold to autoclave heat, and they maintain their integrity even under the harshest of conditions. Now think about Mars. The atmosphere is mostly carbon dioxide, with temperatures ranging from minus 190 degrees Fahrenheit at the poles to a balmy 70 degrees Fahrenheit at the equator in summer. There are global dust storms that can last for months. Any filter system needs to handle these extremes while continuously sampling the atmosphere for potential microorganisms or organic molecules. PTFE membranes are perfect for this. They won't degrade, they won't react with Martian chemistry, and they can capture particles down to 0.1 microns. But the real innovation comes in designing multi-stage filtration cascades, similar to what we use in pharmaceutical water systems, but optimized for life detection. Here's how I would envision one working. Stage 1, a 10-micron pre-filter to remove large particles, ice crystals, dust, debris. This protects your downstream filters and prevents premature fouling. In pharma, we call this the guard filter, and it's crucial for maintaining system performance. Stage 2, a 1-micron depth filter to capture larger microorganisms and aggregates. Earth's largest bacteria can be up to 750 microns, but most are between 0.5 and 5 microns in size. This stage would catch the big stuff while letting smaller microorganisms and molecules pass through. Stage 3, a 0.45 micron membrane filter, the sweet spot for bacterial retention. This is where we'd expect to capture most cellular life forms if they're similar in size to Earth microorganisms. Stage 4, a 0.1 micron filter, or even a 30 kilodalton ultrafiltration membrane to capture viruses, large proteins, and nucleic acids. This is borrowed directly from pharmaceutical virus filtration, where we routinely achieve 6-log reduction of 20 nanometer particles. But here's where it gets really clever: tangential flow filtration, or TFF. In pharmaceutical bioprocessing, we use TFF to concentrate proteins, harvest cells, and perform buffer exchange. The feed stream flows parallel to the membrane surface, with a portion passing through as filtrate, while the retained material stays in circulation. This prevents filter clogging and allows for massive concentration factors. Imagine applying this to an alien ocean sample. You can process hundreds of liters of water, concentrating any microorganisms by a factor of 1,000 or more. That sparse population of maybe 1 cell per liter suddenly becomes 1,000 cells per milliliter, much easier to detect and analyze. The same TFF systems we use to concentrate monoclonal antibodies could concentrate alien microbes. The beauty of pharmaceutical filtration technology is that it's already designed for validation, monitoring, and control. Every filter has integrity testing, bubble point, diffusion rate, pressure hold to ensure it's working properly, has the correct pore sizing, and has no defects. We have established methods for extractables and leachable testing to ensure filters don't contaminate the product. And we can sterilize the entire apparatus to ensure no Earth microbes end up resulting in false positives for results, or even worse, potentially contaminating the entire biosphere of another planet and destroying a pristine alien ecosystem. We understand filter compatibility with different chemicals, temperatures, and pressure. All of this knowledge transfers directly to space applications. Now let's talk about the absolutely wild robots and spacecraft that will carry these filtration systems to other worlds. And trust me, some of these concepts sound like they came straight out of a sci-fi movie pitch meeting. First up, the Europa Clipper, which is currently speeding toward Jupiter. This spacecraft is about the size of a basketball court when its solar panels are fully deployed, and it's packed with 9 scientific instruments. But here's the clever part: instead of orbiting Europa directly, which would expose it to Jupiter's intense radiation that would fry its electronics in weeks, it's going to orbit Jupiter and make 49 close flybys of Europa over 3.5 years. Each flyby will scoop as low as 16 miles above the surface, scanning different regions and building up a complete picture of this ocean world. Now, the Europa Clipper doesn't have filters in the traditional sense, but it's laying the groundwork for future filtration-based missions. It's mapping the ice shell thickness, identifying the warmest regions where the ocean might be closest to the surface, and looking for active plumes like those on Enceladus. It's essentially scouting the territory for the next generation of life-detecting robots. Speaking of the next generation, let me introduce you to the Cryobot. Imagine a nuclear-powered, torpedo-shaped probe that melts its way through Europa's ice shell like a hot knife through butter. Except the butter is 15 miles thick and frozen at minus 260 degrees Fahrenheit. The Cryobot uses radioisotope heating, the same technology that powers many deep space missions, to gradually melt through the ice, potentially taking years to reach the ocean below. But here's where filtration becomes crucial. As the Cryobot descends, it would continuously sample the meltwater, passing it through a series of filters. Any microorganisms living in the ice, and we know from Earth that bacteria can survive in glacial ice for millions of years, would be captured and concentrated. The filters would be periodically analyzed by onboard instruments looking for signs of life. Once the cryobot breaks through into the ocean, it could deploy something even more amazing, an autonomous underwater vehicle equipped with advanced filtration systems. Picture a robotic submarine swimming through an alien ocean, continuously filtering water through specialized membranes. It would be like a pharmaceutical water system crossed with a deep-sea exploration vehicle operating autonomously 500 million miles from Earth. But wait, it gets wilder. NASA's Jet Propulsion Laboratory is developing something called EELS, the Exobiology Extant Life Surveyor. This is literally a snake-like robot designed to slither through the ice fence on Enceladus. It recently completed field tests in the Athabasca Glacier in Canada, successfully navigating through icy crevasses and moulins. EELS could carry miniaturized filtration cartridges, sampling the vapor and ice particles in Enceladus's plumes as it explores these alien ice chimneys. Then there's Dragonfly, headed to Titan in 2028. This nuclear-powered octocopter, that's a helicopter with 8 rotors, will hop across Titan's surface, covering up to 110 miles during its mission. Each hop could take it over Titan's methane lakes and through its hydrocarbon rain. Imagine equipping Dragonfly with specialized filters designed to work in this bizarre environment, capturing organic molecules from Titan's thick atmosphere or sampling the shores of its alien lakes. The filtration systems for Dragonfly would need to be unlike anything we use on Earth. We're talking about filters that work at minus 290 degrees Fahrenheit, that don't dissolve in liquid methane, and that can capture complex organic molecules that might be the precursors to a completely different form of life. It's like designing a filter for a chemistry that doesn't exist on Earth. What makes these missions game-changing is the integration of filtration with other detection methods. You don't just capture material on a filter and hope for the best. These systems would incorporate real-time PCR to amplify and detect nucleic acids, mass spectrometry to identify organic molecules, microscopy to visualize any captured cells, ATP bioluminescence to detect metabolic activity, immunoassays to identify specific proteins or biomolecules. It's essentially taking an entire analytical laboratory and shrinking it down to fit inside a robot operating in the most extreme environments imaginable. The same integrated testing we do in pharmaceutical quality control laboratories, but automated, miniaturized, and radiation hardened for space. Now you might be wondering, so what does a cloud-based manufacturing execution system have to do with finding aliens? Well, let me tell you, it has everything to do with it. Because when you're building equipment that has to work perfectly, the first time, 500 million miles from the nearest repair shop, your manufacturing quality control better be absolutely bulletproof. Let's put this in perspective. In pharmaceutical manufacturing, if a batch fails, it's costly and disappointing, but you can just make another batch. If a filter fails, you can just replace it. If an instrument goes down, you call service. But in space exploration, there are no second chances. There's no service calls to Europa, no replacement parts for a cryobot that's already melted 5 miles into an ice shell. The equipment has to work perfectly, period. This is where cloud-based manufacturing execution system platforms like Apprentice.io's Tempo Manufacturing Cloud become absolutely critical. Imagine tracking every single component of a life detection system from its very beginning, from the polymer pellets that become filter membranes to the final integrated spacecraft ready for launch. We're talking about a complete, unbroken traceability across potentially hundreds of suppliers, multiple countries, and years of manufacturing. And here's how it would work. Every filter membrane starts with raw materials, polymers, solvents, and additives. In Tempo, each lot of raw material has a complete electronic record, certificates of analysis, supplier information, test results, storage conditions. When that material is used to cast a membrane, the process parameters are recorded in real time, temperature profiles, humidity, casting speed, and post-treatment conditions. Every quality test is documented. Pore size distribution, bubble point, bacterial retention, chemical compatibility. But it goes deeper than that. The clean room where the membrane is manufactured has its own digital twin in the MES. Particle counts, microbial monitoring, temperature, humidity, all continuously logged and trended. Any excursion, any anomaly is immediately flagged and then investigated. Because that one particle, that one microbial contamination could potentially compromise a multi-billion-dollar mission. As the filter membrane is assembled into a cartridge, more data layers are added. The housing materials, the seals, the support structures, each component has its own digital genealogy. welding parameters for plastic components, torque specifications for fittings, sterilization cycles. Everything is captured and linked to that specific filter unit. Now multiply this by the hundreds of filters that might go into a single mission. Each one needs to be perfect. Each one needs complete documentation, and they all need to work together as an integrated system. The MES becomes the central nervous system that coordinates this massive effort in manufacturing. But here's where it gets really interesting. The same MES that manages manufacturing can also handle mission data. Imagine the MES receiving real-time telemetry from processing filter differential pressure readings from a cryobot or tracking sample collection data from Dragonfly. The same dashboards that show batch genealogy could show mission parameters. The same trending algorithms that detect manufacturing deviations could identify potential biosignatures. Quality by design takes on a whole new meaning in space applications. You're not just designing for efficacy and safety, you're designing for survival in environments we can barely simulate on Earth. The MES helps manage this complexity through digital design history files that capture every design decision and rationale. Risk assessments that consider failure modes we never encountered before. Validation protocols that push equipment far beyond normal operating conditions. Accelerated aging studies that simulate decades of space radiation. Material compatibilities testing with chemicals that don't exist on Earth. The integrated change control in a cloud-based manufacturing execution system becomes crucial when you're coordinating between NASA, ESA, academic institutions, and private contractors. A design change in a filter housing might impact thermal management, which affects power consumption, which changes mission duration. Every change needs to be evaluated across the entire system, with all stakeholders having visibility. And let's talk about electronic batch records for a moment. In Pharma, we use EBR to ensure consistency and compliance. For space missions, they become the permanent record of humanity's attempt to find life beyond Earth. Every filter manufactured, every test performed, every decision made, it's all preserved in the cloud, accessible to scientists and engineers, not just during the mission, but for decades afterwards when we're analyzing the results. The predictive analytics capabilities of modern MES platforms could be a game changer for space missions. By analyzing manufacturing data patterns, we could predict potential failure modes before launch. Machine learning algorithms could identify subtle correlations between process parameters and long-term performance. The same AI that optimizes pharmaceutical yields could optimize the chances for detecting alien life. Let me tell you about what's happening right now in laboratories and field sites around the world. Because while the Europa Clipper mission is making headlines with its journey to Jupiter, there's incredible work being done right here on Earth to prepare for the next generation of life detection missions. Just this past summer, NASA's Jet Propulsion Laboratory completed field tests on their Orca cryobot prototype at the Juneau ice field in Alaska. This isn't some paper concept, this is actual hardware melting through real ice. They're working out the bugs now in Alaska so they don't have to work them out on Europa. Meanwhile, at the Athabasca Glacier in Canada, the eel's robot snake has been slithering through ice tunnels, testing its ability to navigate and sample in confined spaces. But perhaps the most exciting breakthrough is in microhabitat detection. Scientists have realized that life doesn't distribute evenly, it clusters in favorable spots. On Earth, we see this in everything from biofilms to hydrothermal vents. The same is likely true on other worlds. So instead of random sampling, new techniques use deep UV fluorescence to identify organic hotspots, then precision drilling to extract just those specific regions. This is where pharmaceutical analytical technology really shines. Researchers are adapting the same UV visible spectroscopy we use for protein concentration measurements to identify potential biosignatures on ice surfaces. The same Raman spectroscopy we use for raw material identification can characterize organic molecules in Martian soil. The same mass spectroscopy techniques we use for impurity profiling can identify amino acids in alien ocean samples. One of the most innovative developments is in what's called lab-on-a-chip technology. Imagine taking an entire pharmaceutical QC laboratory and shrinking it down to the size of a smartphone. These devices use microfluidics, tiny channels etched into silicon or polymer substrates to process samples. A single drop of alien ocean water could be split into dozens of parallel analyses. pH, conductivity, organic carbon, amino acids, nucleic acids, lipid, metabolic activity. We just don't know what we're going to find. The filtration components for these chips are absolutely mind-blowing. We're talking about filters with the precision controlled pore sizes down to 10 nanometers, integrated directly into microfluidic channels. They can concentrate samples by factors of 1,000 in minutes using just microliters of fluid. It's like having a complete tangential flow filtration system that fits on your fingernail. Planetary protection protocols have also seen major advances. The paradox of searching for life is that we need to make absolutely sure we're not contaminating other worlds with Earth microbes. So every component needs to be sterilized to levels that make pharmaceutical sterility look casual. We're talking about dry heat sterilization at 125 degrees Celsius for 50 hours or more. Vaporized hydrogen peroxide treatment with multiple cycles. Gamma irradiation at doses that would sterilize anything, and in some cases, complete component bakeout at 500 degrees Celsius. The challenge is sterilizing filters without destroying their functionality. Recent breakthroughs use electron beam irradiation to sterilize assembled filtered cartridges without damaging the membrane structure. It's like performing surgery with a captured lightning bolt, incredibly precise and incredibly powerful. Cross-industry collaboration has exploded in the last few years. Pharmaceutical companies are working with NASA to adapt their technologies for space. Sartorius, Paul, and Millipore Sigma have all contributed expertise to various missions. The same companies ensuring the safety of our medicines are now helping search for alien life. And here's something that really excites me, the development of smart filters with integrated sensors. These aren't just passive membranes, they're actively monitoring what they're capturing. Impedance sensors can detect when cells are retained. Fluorescent markers activate when specific organic molecules are present. pH indicators show metabolic activity. It's like giving the filter its own nervous system. I have my own patents on smart filters and utilizing different communication technologies for getting the sensor data out of the filter assemblies, which could potentially be leveraged here. Alright, let's peer into the future. And I'm not talking about vague hand-waving about possibilities. I'm talking about concrete missions, technologies, and timelines that are already in planning. The future of astrobiology filtration is closer than you might think, and it's absolutely incredible. In the near term, 2030 to 2035, we're going to see the first results from Europa Clipper's reconnaissance mission. By 2030, the spacecraft will be in the Jupiter system, conducting its flybys of Europa. The data it sends back will revolutionize our understanding of this ocean world and directly inform the next generation of life detection missions. We'll know where the ice is thinnest, where plumes might be active, where the ocean chemistry is the most promising for life. By 2034, Dragonfly will be hopping around on Titan, potentially using advanced filtration systems to sample its methane lakes and organic-rich surfaces. But here's the really exciting part. NASA is already planning the follow-up missions. The Enceladus Orbilander, currently in concept development, would arrive at Saturn's moon in the late 2030s. This mission would first orbit Enceladus, flying through its plumes multiple times with sophisticated filter-based collection systems, then land near the South Pole for direct surface sampling. The filtration systems on the Orbilander would be unlike anything we've ever built before. We're talking about filters that can capture particles at velocities of several kilometers per second without destroying them. Aerogel collectors backed by gradient density filters to gradually slow and then capture high-speed ice particles. By the 2040s, I predict we'll see permanent filtration stations established on Europa and Enceladus. These wouldn't be one-shot missions, but long-term monitoring posts. Solar-powered or RTG-powered stations that continuously sample plumes, ice, and any accessible water. The data would stream back to Earth via relay satellites, giving us real-time updates on the search for life. But the real revolution will come from the convergence of AI, robotics, and filtration technologies. Future missions won't just follow pre-programmed sampling protocols, they'll use artificial intelligence to identify promising samples in real time, adjust their filtration strategies based on what they're finding, and even modify their exploration patterns to maximize the chances of detection. Picture an AI-driven cryobot that notices an unusual spectroscopic signature in the meltwater. It automatically adjusts its filter pore size to optimize capture of particles in that specific size range. It modifies the descent rate to spend more time in that layer of ice. It activates additional analytical instruments to characterize what it's found. All of this happens autonomously without waiting for instructions from Earth that would take hours to arrive. The filters themselves will evolve beyond passive membranes. We're already seeing research into bio-inspired filters that mimic the selective permeability of cell membranes. Filters that could actively grab and hold on to specific molecules using engineered binding sites. Filters that can change their properties in response to what they've captured, tightening their pores when they detect organic molecules, for instance. In the far future, 2050 and beyond, I envision something even more ambitious. Networks of interconnected sampling stations across multiple ocean worlds, all using standardized filtration and analytical systems, all feeding that data into a centralized AI that looks for patterns across worlds. Are there common biochemical themes across the solar system? Would potential organisms found on Europa and Enceladus evolve similar life? Are they chiral? All life on Earth is made of right-handed sugars, such as DNA and RNA, and left-handed amino acids to make up proteins. Would we see the same thing on other worlds? These are questions we can only answer with systematic, long-term sampling. But here's what really excites me about the future. The technology we develop for space will transform Earth applications as well. Those ultra-sensitive filters designed to catch a few alien cells, they'll have applications on Earth as well. They could potentially revolutionize early cancer detection, capturing circulating tumor cells from blood samples. The miniaturized analytical systems, they'll enable point-of-care diagnostics in the most remote locations on Earth. The AI-driven sampling strategies, they'll optimize everything from environmental monitoring to pharmaceutical process controls. We're also going to see the emergence of biohybrid filters, membranes that incorporate living cells or enzymes to detect specific biosignatures. Imagine a filter coated with engineered bacteria that glow when they detect amino acids, or embedded enzymes that generate an electrical signal in the presence of DNA. It's bringing together synthetic biology and membrane technology in ways that we're only just beginning to explore. The regulatory landscape will evolve too. Right now, there's no FDA guidance for extraterrestrial life detection devices. But as these missions become more common, we'll need standards. What's the acceptable false positive rate for claiming you found alien life? Beyond seeing something alien wriggling around on the filter membrane, of course. How do we validate a filter that's supposed to work on Europa? These are the questions that will bring together space agencies, regulatory bodies, and the pharmaceutical industry in unprecedented ways. So here we are, at the end of our journey from the clean rooms of pharmaceutical manufacturing to the icy moons of the outer solar system. We've explored how membrane filters, those unsung heroes of drug manufacturing, might be the key to answering the ultimate question: are we alone in the universe? Let's recap this incredible convergence we've witnessed today. The same 0.2 micron filters that ensure the sterility of injectable drugs could one day capture Martian microbes. The same tangential flow filtration systems that concentrate monoclonal antibodies could concentrate alien cells from Europa's ocean. The same PTFE membranes that filter the air in clean rooms could one day sample the plumes of Enceladus. The same quality systems that ensure drug safety could one day ensure mission success in the search for extraterrestrial life. But this is about more than just technology transfer. It's about the profound implications of what we're trying to achieve. Finding life beyond Earth wouldn't just be a scientific discovery. It would be transformational for all of humanity. It would mean that life is not a random accident, but perhaps a cosmic imperative. It would mean in this vast, cold, seemingly empty universe, we are not alone. And even if we don't find life, even if Europa's oceans are sterile, Enceladus's plumes are barren, and Titan's lakes are completely devoid of any biology, that too would be profound. It would make Earth even more precious, life even more remarkable, and our responsibility as its caretakers even more sacred. The message of hope here is multi-layered, just like our filters. First, there's the hope that comes from human ingenuity. Who would have thought that expertise developed to manufacturing safe medicines could contribute to the search for alien life? It shows that knowledge is transferable, that skills developed in one domain could revolutionize another, that the work we do every day, no matter how specialized or technical, could contribute to humanity's greatest adventures. Second, there's the hope that comes from collaboration. The search for life beyond Earth is bringing together people and organizations that rarely interact. Pharmaceutical engineers are working with planetary scientists. Quality assurance professionals are collaborating with astrobiologists. Filtration specialists are designing equipment for NASA. These unexpected partnerships are not just advancing the search for life, they're showing us new ways to solve problems here on Earth. Third, there's the hope that comes from purpose. In our daily work in pharmaceutical manufacturing, we know we're contributing to human health. But now we see that our expertise, our technologies, our ways of thinking can contribute to something even bigger, understanding our place in the universe. So here's my call to action for everyone listening. Whether you're a filtration engineer, a quality professional, a manufacturing expert, or simply someone fascinated in the cosmos, think beyond your immediate application. That filter you're validating, that process you're optimizing, that quality system you're implementing, ask yourself, how could this apply to extreme environments? How could this help explore other worlds? How could this contribute to finding life? Engage with the space community. NASA, ESA, and private space companies are actively seeking expertise from other industries. Your knowledge of membrane materials, your experience with sterile processing, your understanding of analytical methods, these are invaluable to space exploration. Attend conferences, join cross-industry working groups, contribute your expertise. Support science education and outreach. The next generation of scientists and engineers will be the ones with the greatest chance of actually finding life beyond Earth. Support STEM education, mentor young people interested in science, advocate for funding basic science research. The child you inspire today might be the one who discovers alien life tomorrow. Stay curious and stay connected. Follow the missions we've talked about today. When the Europa Clipper reaches Jupiter in 2030, remember that it might be using technologies that you helped develop. When Dragonfly lands on Titan in 2034, think about how filtration is enabling its exploration. When the next breakthrough in membrane technology happens, consider how it might apply to astrobiology. And finally, remember this, we're not just filtering solutions anymore. We're not just ensuring product quality or maintaining sterility. We're part of a larger story, the story of life trying to understand itself, reaching out across the cosmos to see if it's alone or part of a vast community of living worlds. Every membrane we manufacture, every validation we perform, every quality test we run, it's all part of humanity's greater quest. We're not just making filters, we're making the tools that might achieve first contact. We're not just ensuring compliance, we're ensuring that when we claim to have found life beyond Earth, we can back it up with data that would satisfy even the strictest regulatory review. The universe is vast, cold, and mostly empty, but in that emptiness, on at least one small blue world, matter has organized itself into beings capable of wonder, of curiosity, of reaching for the stars. And now, armed with filters and determination, we're reaching out to see if we have cosmic company. Whether we end up finding life or not, the search itself ennobles us. It represents the best of human nature, our curiosity, our ingenuity, our refusal to accept the unknown as unknowable. And in that search, the humble filter, that simple device for separation, becomes an instrument of connection, a bridge between worlds, a tool for answering the deepest questions that we can ask. We're not just filtering for solutions, my friends. We're filtering for our place in the universe. And that makes every one of us, every filtration engineer, every quality professional, every person who's ever validated a membrane or run a bubble point test, part of the most profound adventure in human history. In an industry where precision and compliance are paramount, Apprentice's Tempo platform offers a cutting-edge manufacturing execution system solution. It's cloud-based, mobile-first, operator-centric, with the most intuitive UI in the industry, making it perfect for today's dynamic pharmaceutical manufacturing environments and for the production of the world's most complex drug products. Whether you're working on groundbreaking cell and gene therapies, biologics, or small molecules, Tempo provides a fully validated MES system that's both powerful and easy to use. It streamlines your processes, ensures compliance, and adapts to your specific manufacturing needs. So get off of paper batch records and discover how Apprentice's Tempo Manufacturing Cloud can transform your operations by visiting us at apprentice.io. Attention industry professionals, revolutionize your manufacturing process with the Apprentice Tempo Manufacturing Cloud, a manufacturing execution system built to streamline your operations, boost efficiency, and ensure compliance all in one powerful platform. Experience the future of manufacturing with our free 30-day trial. Scan the QR code on your screen now to unlock a world of innovation. Don't just keep pace, set the tempo as an industry leader with Apprentice. That's all for Apprentice.io's Industry Insights. I'm Frank Maggiore. Thanks for listening. Be sure to like and subscribe. Hit the bell icon for notifications when new episodes drop. Please comment on if you have any additional topics you would like for me to cover in this space, and we'll see you next week for more Industry Insights.